Welcome back to Bioinformatics with BB channel. My name is Babajan Banganpalli and I am an associate professor in bioinformatics. On this channel, we cover about various bioinformatics or biological data analysis concepts in genomics, proteomics, and metabolics. So if you like this content, please subscribe our channel. Today, we are going to learn about homology modeling. Before we enter into the hands-on section on the homology modeling, I just want to give you a quick 1-minute introduction to the homology modeling. Let's start. Protein molecules are a class of chemicals in living organisms that are helpful to maintain the life of the organism by carry out various biological activities. It is very important to understand the structure of a protein if you want to know the function of a protein. If you don't know the structure, it's very hard to understand what exactly the function of a protein. So most of you know that protein structures can be solved by the experimental methods, X-ray or NMR or electron microscopy method. By using three different methods, we can solve the structure of the proteins. But if we take the disadvantages of these technologies, they are really highly expensive and requires it needs so many resources are needed, and also it's going to take a lot of time. It's time consuming methods. So apart from that, they are not that much successful with all the proteins, especially if you are working on the membrane protein. It's very hard to determine the structures of the membrane proteins. So if you say the Uniport or Trimble or Swissport, these databases, nearly 180 million protein sequences are deposited in this Trimble. Okay, so among them, only 0.16%. You can see that the RCSP1, the protein data bank 1, only 0.16 million proteins have their structure solved experimentally. So although recently the rate of experimental structures determination will be continuously increasing, but the number of newly discovered sequence grows much faster than the number of structures solved. As we know, nowadays we have so much of high throughput technologies are available, where it is allowing us to sequence a huge amount of biological data. You can see the huge gap. Here we have 180 million sequences are there. Here we have only 0.16 million is there. There is a huge gap is there. There is a huge gap between the number of available sequence and experimental solved structures. This gap could be resolved by the computational methods. Okay, so computational method, there are two methods are available if you want to do the computational analysis to develop a protein structure. One is the ab initio method, and the second one is a homology modeling method. So I'm not going to focus on the ab initio method. Why means next video we are going to more in details, we are going to work on the ab initio methods. So let us talk about the homology modeling. So what is homology modeling means? Homology or comparative modeling. Homology modeling or comparative modeling is a technique to create a three-dimensional structure of a protein in atomic details on the basis of the known experimental structures. Okay, so you have to remember this is only exclusively possible if your sequence has been at least 45% identity with the already known structure. Okay, and your known structure should be resolved by experimentally, I mean either by any one of the method like X-ray, NMR or electron microscopy. It should be resolved by these methods, otherwise you cannot work with this homology model. Okay, so here there are so many softwares are there. I'm going to provide a link for you, just check in the description box. There are so many softwares are available to perform the homology model. Okay, so out of the all the softwares and web servers, we always prefer to use the modeler. Why means they use highly successful spatial restraint methods. So the modeler not only provides you a homology modeling structure, it also gives you a few other options, like if you want to optimize the structure, if you want to create a loop model, if you want to optimize the loop model. Okay, so if you want to check the quality of the model, so we can do so many things by using the modeler tool. Okay, so here if you want to install the modeler, it's very easy. So you need to register modeler. I just provided the link in the description bar. Just go through it. You have to install the modeler. You need to get a key. For academic people, uh, it's a free. If you just need to register modeler, immediately you're going to receive an email. So it will support Windows, Mac, and Linux. And but you must need to install the Python 2.4, minimum of Python 2.4 you have to install. So if you see what are the major files are needed for the modeler, there are two major files are needed. One is alignment file, and the second one is a template file in protein data bank format. So let us see what are the steps are there in homology modeling. So there are various steps are there. The first one is we need to get a amino acid sequence in FASTA format. So we will get this sequence from various databases, like you can get this from Uniport, NCBI, Ensemble, Keg. You can get from anywhere, but they need to have a FASTA format. So once you got the sequence, we need to identify what are the identical protein data bank templates are available. So we can do these steps by using the BLASTP. So okay, by using a NCBI BLASTP, we will identify which template is having a high identity with our target sequence. Once we identify our template, so we're going to perform the pairwise sequence alignment or multiple sequence alignment with our target sequence. So pairwise means if you are using the one template, then we call that one as a pairwise sequence alignment. If you are using the multiple template, we call that one as a multiple sequence alignment. So in homology modeling, it's always preferable to use a multiple template to build the accurate models. So once a model has been generated by using the modeler software, we have to validate that one whether the quality of the protein is accurate or not. We will take an example of a COVID-19 nucleocapsid phosphoprotein to develop a protein structure by homology modeling method. Let's start. Okay, in first step, we need to get a protein sequence in a FASTA format. So you can get this sequence from anywhere, from Uniprot, Ensemble, or NCBI. So all these databases will provide you the information for you. So now I'm going to get this information. I'm going to get this FASTA sequence from NCBI. Okay, so this is a protein I want from COVID nucleocapsid phosphoprotein. Okay, just I'm clicking this one. So here in right side panel you will see the option called as a protein. If you want a nucleic acid sequence, you have to click the FASTA here. So here now we need a protein sequence, so click on the protein. So here you need to get this full form, okay, not the isoform. Okay, here nucleocapsid phosphoprotein severe acute respiratory syndrome coronavirus 2. Click this one, and here it will show you information about the sequence. So we need this sequence in a FASTA format. Click on FASTA. So here you copy this sequence information. Okay, so here I'm creating one folder here, COVID-19. Okay, so here again I'm creating one text document here, okay. So here np, so paste the sequence. So remove this part, So remove this one, we label this one as a target. So save this one. So now we need to find out the templates. What are the identical regions, what are the known experimental structures are available for this sequence? Simply, you can just click on the run blast here, or if you want to run from external side, you have to click blast p in a Google. You will get the same website, same NCB file. You will redirect to the same website. So here I'm just going to click this run blast. Okay, so here in database, so we are looking for a structure, then you have to select the protein data bank. So show results in the other tab, click next. So see here, it will show you so much of information for you. Okay, so here 490, this is your query sequence. Once you load this one, it will try to align with the already available information in database. It see here, it found 23 selected sequences are there. So you can see what is the identity is there. So nearly 99.28% identity is there. So the coverage score is 32%. It means out of 100 amino acid, it covered 32. So for example, if you have more than 420 amino acids are there for this sequence, only 32% is covered. Okay, you can visualize this information in a graphical manner. You can see here, click on graphical summary. It will display you various color for you. If your match is more than 200 amino acids, it will show you this color. If it is match is less than that one, it will show you this color. Sorry, if the match of alignment scores, if it is more than 200%, it will show a red color for you. And if it is less than 80 to 200, in between 80 to 200, it will show you this kind of colors for you. So here out of all the templates available in the protein data bank, you can see here for the coronavirus 2 NP protein, there is no full length structure is available. So if you look at this structure, if you look onto this sequence, this is your sequence from 1 to 420 amino acids. So only partial form of the crystal structure has been done, not the full length form. Okay, so now we need to consider these templates to build the full length of NP protein. Okay, so we are going to take any you can take any one of them. So you can see this red score, it determines it's a high frequency, high alignment scores. So I'm going to take this sequence and this sequence and this one. So we are going to use a multi-template approach to develop the protein model. Okay, so first we are going to, for this we need to get the two things. One, we need a sequence of template, and another we need a structure of template. First we'll get the sequence of this template, and also we need a sequence where exactly the match is there with the target sequence. Okay, so you can see here only this region. I don't want the full sequence of this protein, so only the region where exactly the crystal structure has been solved, that region only I need. Okay, this one, click here, alignment. Okay, so if you click on the alignment, it will show you that the protein ID name for you. Okay, so I'm just going to copy this one, download, store here, align file. I'm going to save that file in COVID. Okay, you have to relabel them, otherwise you will get confused. So here 6M3M is a sequence of 6M3M. Okay, if you open that region, it is not going to show you a full length for you. Okay, only the partial part of the sequence has been, whatever the sequence which is mapped with the target one, only that region has been shown. Okay, so here this is a sequence of 6M31. So I'm going to take one more region. So just go back to the graphic summary one more time. Okay, see here, this region is matching this sequence we have considered. It is matching from approximately from 47 to 48 base pair to nearly 150 or 170 base pair. You can see here second template, even second protein structure. second PDB structure, it is somewhere it is matching from 240 amino acids to nearly 350 amino acids. Okay, so here we have a two different template, two different PDB structures are available. We have to combine them, and whatever the missing parts are there, it is going to covered by the modeler. Okay, so here click this one, alignment. So this is the region, click here, download. So here, okay, so as I told you, you have to label them properly, otherwise you will get confused. So now this one is structure, this PDB. So here remove this part, so we don't want all this information. So now we have a three protein sequence. One is unknown, I mean target one, which is NP, and these are the partially crystallized, partially not a full form, partially crystallized structure. So as I told you, apart from the sequences, we also need the crystal structure. So to get the crystal structure, you have to go to RCSP. Type the ID. Okay, here, download. PDB, save link. So here I'm going to save this one, the same folder. Okay, here COVID19 PDB. So the PDB has been saved. Similarly, you have to do with the other model also, 6M, 3M. Download PDB, save link. Okay, so here we save this one as the PDB file. Okay, so one more thing. Now we have the sequences and PDB structure. So one more thing we have to do the manually. We need to edit the PDB structure. If you check this PDB structure, consists of various chains. You can see here A, B, C, D. Various chains are available. But here, your chain 6M, 3M has been mapped with the chain A only, not the other chain. So we have to remove the other chains along with the heteroatom. There are whenever the crystal structure has been formed, it has been built with the different kinds of water molecule, different kinds of heteroatoms. If you add these heteroatoms to your PDB structure or crystal structure, the program modeler, it may not able to recognize that one. So to do that one, we have to do the manual creation. Even for the other protein also, even for so this one also A chain is matched. Okay, we have to remove other structures or other regions. So first look for the A chain. So you can, how you can open this one, just to you have to go to the open with, and word pair or notepad, you can use anything. So here chain A is starting from, so here 49 amino acid. You can see it's just drag down. So here you can see end of this chain 174. Now I'm going to remove the other chains. Okay, just click on the first atom of the other chains, hold the shift, click on the last atom, don't remove this end. Okay, so delete this one. Okay, you can find only this region. Now save. So do the same thing for the other protein also, other crystal structure also. So here I'm going to take only chain A. Chain A. Okay, so we have to take only chain A. So other chains will remove the other chains. So here, so select the first atom, drag down to end. Okay, so we have to place this in. So save. Now we have a sequence and structures. Okay, now we have a structures and sequence. So next step is we need to align the sequence. We need to align the amino acid sequence with the known sequences. Okay, so to do that one, first you have to open the target one. Okay, so target one, open the target one. and open the templates, copy this region, paste in one file. So here pasting these two things here. Okay, so we'll remove this part. So now save this one. Now we need to perform the alignment. Okay, so we need to perform the alignment to check what exactly the identical regions are there in between the target and the template. In between the target, and you have to remember, in between the target and template, not in between the template and template. Okay, so we are going to map, we are going to align the two sequences. We have performed the multiple alignment, so sorry, multiple template modeling. So if you are performing only one model, it's a pairway is more easy for you. If you are using the multiple templates, if you are using the multiple template, it's slightly tricky. You have to use this alignment in different format. Okay, so I will show you how to do that one in a simple way. Okay, next one is we need to align the sequence. We need to generate a PIR format file. This file we can generate by using the program called as a ClustalW XPASI ClustalW. Okay, so here you will find the ClustalW server. So here you have to use PIR output format option. Now you have to use, as I told you, if you give all these three sequence in a one slot to the ClustalW, what it will do is it will try to match, it will try to do a multiple sequence alignment with all the sequences. Okay, even it will compare this sequence the structure, sorry, template structure with the template structure, but we don't want that one. We want to compare this sequence with the target one. Okay, so at that condition what we need to do is, so first thing we have to select the one target and one template, target and template. Okay, so copy this one, paste here, run for output. So here click on PIR format. So here I'm going to save this one, open this one, so open with the same word copy. Now you can see here it has been aligned. It is showing this region. Okay, this whatever this dashed regions are, it means they are the missing regions. These are the missing regions are there. So now we align with the one sequence. The next step is we have to align the same target sequence with the same target sequence with the other template. Okay, so here I'm going to do the alignment with this one. So go back. So now in this case you need not to copy the target one. Okay, so here you can copy only the template one. Okay, so now we are going to paste this one here. Okay, now you can see here this is a target region and these are the two template regions. In one template region their matching is 0, so if the residue is available, it means it has been matched with the target sequence. If it is dashes are there like this, that these symbols are there, it means there is a gap, there is no sequences available in the target sequence. Okay, so you have to remember this one. So once you done this one, go to file, save as. Okay, so here we're going to COVID, type as 1.ali. okay, alignment format file. So click S. Now we have everything. So now create one more folder here like raw data. Okay, raw data. So we want only a crystal structures. Okay, we want PDB structures and the alignment file. Okay, we don't want other structures, other format files. Okay, so now we have the alignment file and PDB crystal structure. Now we are ready to start the modeling actual modeling step.